Now, we have seen what a nucleotide is, and we have said that a nucleotide is made up of let's name them together. Made up of, yes, a phosphate group, okay. We have seen it is made up of an organic base, and finally a pentose sugar. Now let's talk about each and see.
Why don't you say nitrogenous base?
I said an organic base or nitrogenous base. It's all the same thing. Nitrogenous base. It's all the same. So now, let's talk about each and see what actually these ones are. And we shall start with a pentose sugar. And this pentose sugar is simply a carbohydrate that is made up of 5 carbon atoms. That's why it is called a pentose sugar. Don't forget your chemicals of life. We are going to use it so much. Okay? There are 5 carbon atoms arranged in a pentameric ring. In a ring to form, it's like a pentagon. Okay? A pentagon. And at each corner, apart from here, is a carbon atom. So it occurs in 2 types. It's either a deoxyribose sugar, and it comes from D means like removal, oxy, so it is removal of oxygen or oxygen removal. So a deoxyribose sugar, it's a ribose sugar that has 1 oxygen atom less than a ribose sugar, and the difference is simply on carbon number 2, whereby on carbon number 2, now this is carbon 1, this is carbon 2, this is carbon number 3, this is carbon number 4, and simply 5. Now on carbon number 2, for a deoxyribose sugar, it just simply have hydrogen and hydrogen. But for a ribose sugar, instead of hydrogen, it has the oxygen. And basically that's the difference. That's how different these 2 sugars are. And for you may think, this is some tiny minor difference, but actually this difference is enough to create a big difference in the 2 molecules formed by these 2 sugars. It makes DNA to be DNA and RNA to be RNA. And DNA performs its own, and RNA also has its own function. Okay? So you may feel this is some minor difference, but actually it's not minor. It is some big difference. So this is a deoxyribose sugar, and this one is a ribose sugar on carbon number 2. So there are 2 types of nucleic acid depending on the pentose sugar, where we have the DNA and the RNA, whereby the deoxyribonucleic acid has a deoxyribose sugar and a ribonucleic acid has a ribose sugar. So that's how cool, that's how simple this work is. Now for organic bases, these ones, they are actually kinda perplex, and this is a caution. You don't need to cram this structure, but there are some few basic concepts you should have, and it's what I'm going to give you. At this level, you don't need to cram these structures. So organic bases, they are heterocyclic compounds that are made up of carbon, hydrogen, and nitrogen within the site. Carbon and nitrogen within the compound, within the site. Now there are those that are made up of two rings, and there are those that are made up of just a single ring. And by the way, these organic bases, they are the ones that are actually most important as far as determining a particular character of a nucleotide is concerned. These organic bases, they are the ones that actually make what we call a gene. Depending on their sequence. We are going to see all that, so have no worries. Okay? Depending on their sequence, it determines a given sequence of a gene. So you can have a look. These are my organic bases. You have nitrogen, you have nitrogen, hydrogen, nitrogen, hydrogen. So these nitrogen hydrogens you are seeing, they are the ones that can actually bond with other nitrogen hydrogens or bond with a OH on carbon 1 of a pentose sugar to add that organic base to form a nucleotide. So they do determine the character of a given nucleotide. And yes, we have seen that this nucleic acid, it's just arrangement. It's just an arrangement of these building blocks called nucleotides. But actually, guys, it is something more than that. Yes, we said that the nucleotide is the nucleic acid, or these nucleic acids are just a sequence of nucleotides added together to form such a strand. But mind you, they are not simply added. It's not a matter of adding. Like when you are building that house that you are in. It's not just a matter of adding, no. They are added in a coordinated manner so that you maintain the sequence. When the other diagram, you saw that these organic bases, they oriented outwards, not so? There. Then you have the phosphate sugar backbone. Okay, the phosphate sugar backbone. That's how it was, not so? Where you had a phosphate group here and another phosphate group here binding the two nucleotides. Okay, that's how it was. And these organic bases, they were oriented inwards. That's what you saw, right? That's brilliant. So this is a basic, if we are to compare just this, that is a basic structure of a nucleotide. Sorry? Just this, this is a nucleotide. Now, these nucleotides are not simply added there. They are not just added, but rather they are added in a coordinated manner so that you come up with a given sequence of these organic bases. And that sequence must capital letters, bold, red, must be maintained. Okay? That sequence of organic bases must be maintained. And we have seen that we have two types of organic bases. There are two types, the purines and the pyrimidines. The purines and the pyrimidines. And we are going to talk about each. We are going to talk about the purines and the pyrimidines. Now, naturally, there are there are 5 known organic bases. There are 5 known organic bases. That is, we have base adenine. This one is found in ATP also. Adenine. We have base thiamine. Hi. Do we have A in the spelling? We shall fact check when we reach there. Then we have base guanine. Guanine. We have base cytosine. And we have base uracil. So those are the 5 organic bases that are known. 5. And usually they are abbreviated using letters, where adenine is abbreviated as A, thiamine is abbreviated as T. Now these letters are known internationally. They are standard. Guanine is abbreviated as G, cytosine is abbreviated as C, and uracil is abbreviated as U. So these are the 5 organic bases that are known. But these 5 organic bases, they are into 2 types. They can be categorized into 2 types. And the 2 types are, they can be purines or they can be pyrimidines. So now, you should be asking yourself that what are purines? Of these 5, which bases are purines and which bases are pyrimidines? Let's talk about purines. Nitrogen in the rings, they are in the ring in their basic structure. You can see that. So now with purines, there are 2 of them. We have base adenine and base guanine. Those are the 2 purines that we have. And they have their double structure. They have the double ring structure, okay, comprising of a hexose sugar, a 6-carbon sugar, okay, and a pentose ring or a 6-carbon ring. That is base. So those are the 2 double rings. In fact, it is not, it's a hexose ring, not sugar. This is supposed to be a ring, okay? It is hexose ring, and then ring. So it is a ring made up of 6, and then another ring that is made up of 5 sides. So it is a pentameric ring. Oh, my pen is misbehaving. And they are of 2 types. Base adenine, base adenine, and base guanine. Now, how are you going to, now you're supposed to know that. You're supposed to know that. Because I'll give you an objective question, and I say which one of the following bases are purines. Which one of the following bases are pyrimidines? and I give you A, adenine. B, I give you A, adenine and thiamine. B, adenine and guanine. C, adenine and cytosine. D. And then you have to choose which one is a purine, which one is a pyrimidine. So I managed to develop some simple acronym, some simple that can help you recall, that can help you easily recall which one is a purine and which one is a pyrimidine. And it is just, if I ask which one is a purine, just remember this: pure silver. And silver, in the chemical formula of silver is this. Okay? So purines, just pure silver. And you know that purine, pure, it is for purine. Okay? And purines, I have base adenine and base guanine. Okay? So pure silver, base adenine and base guanine. Now the rest out of 5, they are all pyrimidines. And so what are pyrimidines? For them, they have just one single hexose ring, one single ring. Okay? Either it is heterocyclic ring, you can see, you have nitrogen in addition to. And they include cytosine, thiamine, and uracil. And obviously, cells will always continuously produce these organic bases, because at any time t, the cells will need to replicate, so they will need to double the DNA, they will need to carry out protein synthesis, so they will always continuously produce these nucleotides, so that you have a pool of nucleotides from which this is supposed to be P double O. You have a pool of nucleotides from which nucleotides can be used whenever required. Otherwise, you don't need to first, let's say you want to carry out replication, you don't need to first synthesize nucleotide with A so that you add it there. You synthesize another nucleotide with C, and then another one with G, another one with A. No. You must have a pool so that when you want to add A, then you just get A and add it there. When you want to add T, you just get T and add it there. So that you form that, you maintain that order, that sequence of amino acids. Okay? You maintain that sequence of amino acids. Otherwise, if you don't, then it becomes berserk. So with pyrimidines, we have cytosine, we have cytosine, not cystosine, thiamine, which is represented as capital T, and uracil, which is represented as capital U.